All right. So today we are doing the starting the nervous system. So the last bits that we're doing, nervous system and endocrine system. So notice that takes up 3 whole weeks in and of itself. So it's a lot of information. And even though it's a lot of information, what I don't like about it is a lot of it's like very surface level. So because we don't have the time to get into the details of it. So we will do the best we can. I just looked at the 3rd exam, and I'll make sure I hit those highlights as we go along. Questions related to this. It is freaking hot. All right, so starting with the nervous system, embryonic organization, we all start as a tube, right? A neural tube. And those tubes have sections, anterior, middle, and posterior. And then those subdivide into sections that will eventually become our cerebrum, midbrain, pons, all those structures, okay? At some point in time, secondary vesicle stage, where we are actually getting into subdividing into our more permanent, what are going to become our more permanent structures, we have the telencephalon, which becomes our cerebral hemispheres, okay? Now, the reason why I'm going over this is, 1, it's part of your IOs, and 2, sometimes people refer to the cerebral cortex as a telencephalon, okay? Diencephalon, that didn't change. It means two-brained. It's basically subcortical structure. They didn't come up with a better term for it, so they just left it diencephalon, whatever. Mesencephalon, middle brain, that becomes the midbrain. Then, of course, everything below that becomes the pons, medulla, and the spinal cord. In fact, myeloencephalon means associated with spinal cord, essentially, rough translation. Okay, so be aware of the different embryonic structures and what they will become. So the important columns here are these two. All right. As far as the organization of our nervous system, we have a spinal cord, and then let's say this is the brain. All right. Those two together are called the what? CNS, central nervous system. All right. And the way this works is we have sensory fibers running in to the spinal cord. They run up to the CNS, they come back down from the CNS, and they come out the spinal cord into the periphery. All right? You're going to find later on that this is like this drawing is almost a lie, right? And we'll find, and if you know anything about the nervous system, you'll know why. But the point is, we have sensory information going in, it gets processed, and then we have motor information coming out. And that's what these diagrams are trying to show you. When we talk about information going in, up, and back, what are we really talking about? You guys had the first 2 weeks lecture, you had some neuro stuff, right? What are those things that are going in, up, and back down and out? Axons, right? They are axons. So it's our axons connected to different receptors and connecting with each other that allow this information to be relayed. So that's an important component of this. Really, we're just a bunch of wires, okay? In a tube. Yes, don't forget about the tube where we can. All right, 3 major levels: spinal cord level, lower brain level, and higher brain, or what we refer to as the cortical level, higher brain functions. At the spinal cord level, here we say it's a bunch of axons running up and down to allow the periphery to communicate with the brain. But if you recall reflexes can go in and then come right back out, right? Without any brain input. Okay, so there are circuits that allow us to keep ourselves from danger, and those are called withdrawal circuits. You ever heard that old saying, like a chicken with its head cut off? Why can a chicken run around with a chicken? It doesn't run with a chicken. It runs without its head, okay? Because walking circuits are located within your spinal cord. So it doesn't need a brain in order to actually walk around. That's why it's able to do that and not just falling over, okay? Gravity circuits, in other words, keeping us upright, posture, as well as reflex control of organ function. So not just associated with like what we think of as muscles, what we can see, but in our organs as well. And we talked a little about that when we did all the other organ systems. All right, lower brain level, that would be this area right here. Before we get to the cortex, medulla, pons, all of these structures right here are so part of the lower brain level. So this is stuff that happens that we don't have to think about, okay? Because if we had to think about breathing, we wouldn't be sitting here, I think I've said this before, we wouldn't be sitting here listening to lectures, right? We wouldn't be able to take that information in. So these are just a small list of some of the things that happen within those lower brain levels that we don't have to think about. And then, of course, the higher brain levels. Our thinking, our ability to reason, our ability to recognize, associate things, think, okay? Now, this is an important statement. The cortex never functions alone. It always needs some sort of input from the rest of your body. In fact, if you really sit there and think about it, most of the memories that come up in your brain were triggered by something that you are doing, looking at, hearing, okay? So we need sensory input for these from lower structures, spinal cord, periphery, lower brain centers, for the cortex to function properly, okay? We know it's got a big hard drive, okay? In fact, I just listened to a podcast the other day that talked about how complex the brain actually is and how little we really understand of it. Okay, and it's what specifically makes us human, okay? This is where our consciousness resides, whatever that is, and we still don't know what that is. But there are good books out there about it. Okay, so really what this boils down to, instead of just a quick in and out, we're like a computer. I think of us like we have a central processing unit, we have a hard drive, we have wires that are connecting each, we have a monitor, we have keyboards, okay? So basically just a complex, I'm not going to test you on this, just an association. All right, now, all of these structures are living, right? And if we're living, we need oxygen, nutrients, and we need to get rid of waste. We also need hormones to be able to interact. So we have a very important anatomical structure that allows for adequate cerebral blood flow. Now, your cerebral blood flow comes from two sources, your internal carotid arteries and your vertebral arteries, okay? And what happens with this structure, which is called the circle of Willis, you guys are probably too young. What you talking about, Willis? You guys know that? Okay. What you talking about, Willis? That will allow, let's say, for example, if one of our internal carotids goes out, we are still able to circulate blood to all parts of the brain because we have this circle that is continuous, allows continuous blood flow. Okay, that's the purpose of the circle of Willis. All right, in anatomy, we talked about all the different labels of these. I don't know if you guys have gotten to that yet, but we're not going to go into the structure of that here. But just as a reminder, these are what these arteries are. Okay. Why do we have this safety mechanism? Because the brain is highly metabolically active, all right? And it doesn't rely at all on any anaerobic metabolism. which means it needs an adequate blood supply at all times. In fact, if you cut off all blood supply to the brain, you pass out in 5 seconds, 5 to 10 seconds. That's going to depend on the person, but that's how important this is. All right, what this slide shows you, let's go to the pictures first. Actually, let's go back for a second. Anterior cerebral arteries, middle cerebral arteries, posterior cerebral arteries. These are the areas of the brain that get supplied by those arteries. So everything in yellow right here would be the anterior cerebral artery. Everything, and notice that's in the middle of the brain between the hemispheres. Everything that's middle cerebral artery is here, and everything that's posterior is in blue here. So I have the key right here so you guys remember. So we're getting ahead of ourselves. What does the occipital region do? Vision. Visual, specifically visual processing. Okay? So if we have a stroke in our posterior cerebral artery, meaning we don't have blood supply going to the posterior cerebral arteries, how would we know that? Vision goes, right? So you can tell which, as a provider, which artery is affected by the manifestations, okay? And that'll tell you what part of the brain is affected and the artery. All right, just a few numbers I won't test you on, showing that the cerebral blood flow and brain is very active and it's tightly regulated. Okay, so we need to make sure that we have an adequate blood supply to meet the brain's needs. Okay, now there are imaging techniques we can use that will look at cerebral blood flow. And the theory behind this is that if you have a region that is more active, it's going to receive a higher amount of blood flow because it needs more oxygen. Okay, does that make sense? So when you are doing these studies, and let's say you have your subject looking at something and just looking at pictures, what part would you see lit up? What if you have them listening to music? Temporal lobes, okay, would light up. All right, they did this experiment, I think this is in cats, where they looked at blood flow to the occipital region, and notice blood flow is constant. Then you shine light in the cat's eyes. They love that, right? And as soon as you shine light in the cat's eyes, what happens to blood flow in the occipital region? It goes up. Then, of course, you stop, and then what happens? Blood flow goes back down. So you regulate blood flow according to need, which is why they use these cerebral blood flow as a measure of brain activity in certain areas. All right? Question for you guys. We know that in the spinal cord going up and down and out, those are axons. But what are axons coming off of? All right, before that. What are axons in our brain and spinal cord? Gray matter or white matter? They are the white matter. All right, what are all our cell bodies and dendrites? Gray matter. So my question to you is, if I measure blood flow in gray matter versus white matter, which is greater? What did you say?
I said gray matter.
Gray matter, because that's where the cell bodies are. That's where the life of the cell is. That's where all of the action is taking place in the processing. Bless you. All right. Now, we know we have glial cells as well. We're not going to go through all of those glial cells, but I do want to mention astrocytes. And astrocytes, if you look at them, they're these green cells right here, and they have appendages that go around the capillaries within the central nervous system. And they also are able to receive neurotransmitters. Okay, so what that tells you is that based on the activity that's going on in an area of the brain, you'll stimulate astrocytes to cause vasodilation in that area, which will bring more blood to that area. So there is a very specific mechanism that is tied to that increase in cerebral blood flow based on activity. And the way this works, so, you know, I'm explaining all of this. I need to get an eraser in here. All right, I know. There's your axon. Let's say this is a dendrite. And here's our astrocyte, okay? Now let's say you have this neuron being active, and it's going to release a bunch of neurotransmitter into the synaptic cleft. And we know that's going to bind to receptors, send a signal down this pathway. But if you have that active so much that a little bit of neurotransmitter leaks out of the synaptic cleft, right? This is something you don't really learn in normal anatomy and physiology undergrad. It will bind to receptors on the astrocyte. Then the astrocyte will become active and cause a wave of calcium to go through, going to its foot that surrounds the capillary and causing vasodilation because of the activity that's occurring in neurons, okay? Oops. So that takes us to the blood-brain barrier. Now, what I'm giving you as the blood-brain barrier is less complex than what the blood-brain barrier actually is. So for our purposes, the blood-brain barrier is a capillary phenomenon that exists only in the central nervous system. Okay, capillary phenomenon that exists only in the central nervous system. All right, so we're talking about the capillary endothelial cells. What it does is it prevents substances that you don't want in the brain from getting into the brain. Okay, so why does it do that? Well, what do we know about capillaries in the rest of our body? Good, they're leaky, right? They're leaky because you want exchange to occur. And where are they typically leaky? Between endothelial cells, right? Well, in the blood-brain barrier, you have endothelial cells that are attached to each other by tight junctions. And because they're attached to each other by tight junctions, if you look at this, let's look at the top of this endothelial cell. And here's another one, and here's another one. They are literally sewn together by these tight junctions, which prevents anything from getting between them. So if you want to get across the endothelial cell into the central nervous system, you have to go through the cell. And what do we know about going through the cell? What is the cell usually permeable to? Lipids and gases. Okay, lipids and gases. What you also should know about the endothelial cells is there is a certain kind of lipid in the cell membranes of the endothelial cells that you won't find anywhere else in the body. And typically those are sphingolipids. Okay, we're not going to go into the cell biology of that, but you'll find those in the brain and nowhere else. And you'll see a large concentration of, what is that? Cholesterol, which makes the brains, the cell membranes, less fluid. Okay. So only lipid stuff can get through. You have specialized carriers in the cell membrane that will only allow certain things through and not other things through, so thus preventing anything water soluble from getting in unless the brain wants it there. If things potentially could get into the blood through the blood-brain barrier, but it doesn't want that, you also have an enzymatic barrier, so there are enzymes on the surface of these endothelial cells that will break things down before they could have an effect on the brain. And let's say something lipid soluble gets into the blood through the blood-brain barrier and we don't want it there. What these cells will do is pump that substance back out. So that's an efflux pump. Pump it back out into the blood because nope, we don't want that there. Okay, so all of that contributes to the blood-brain barrier. And notice, it's not in the brain at all. It's in the capillary endothelial cells. Okay? So, if we want to design a drug, and that's these purple boxes at the bottom, that will get into the brain, it needs to be lipid soluble, and it has to avoid those efflux pumps. All right, so that's an important thing to consider when designing drugs that affect the brain. All right, I'm only going to say about this slide is that you have protective coverings. How many protective coverings? 4. Why do I say 4? Because the skull is a protective covering as well. Skull, dura mater, arachnoid mater in the subarachnoid space, and pia mater. Now, what's the most protective of those? Skull. What's the second most protective? Dura mater. Dura mater means tough mother. So think about it this way. Have you guys ever seen real dura mater before? If you've done gross anatomy dissections, you probably have. Normal membranes in our body, not very protective. You can rip them apart very easily. But dura mater is made up of dense regular connective tissue, much harder to tear apart. So it is a very protective. I remembered to bring it today. I'm proud of myself. So, very protective, very protective mechanism with this dura mater. So that's more of an anatomy thing, so I don't want to spend a whole lot of time on it. Within the arachnoid mater, you have cerebral spinal fluid. And since this is the physiology portion of this, you have to know the function of cerebral spinal fluid. And that's basically, whoa, that would have been bad, to float the brain. Literally, our brain is sitting on a thin bed of fluid. All right. In fact, only about 150 milliliters is what protects our brain from running into hard structures, which would be the skull. Never mind. Okay. What you do have to know, though, is that cerebral spinal fluid, so the purpose of that is to not just make the brain float, but it's a medium for substance transfer, nutrient delivery, waste, getting rid of waste, as well as hormonal flow. So, where is it formed? Within the lateral ventricles, you have these choroid plexuses. They're just a network of specialized blood vessels that produce cerebral spinal fluid. Those choroid plexuses secrete the fluid, fills up the ventricles. It goes, and these are all numbered 1 through 8, so they're very easy to follow. Goes through the interventricular foramen of Monroe. Okay, then it enters the third ventricle, which is right here around the thalamus, hypothalamus. Then it goes through the cerebral aqueduct of Sylvius, okay, down into the fourth ventricle, which is associated with the cerebellum. And then some of it goes down into the central canal of the spinal cord. Others go through the median aperture just below the cerebellum and go through the posterior portion of the spinal cord and around through the posterior part of the brain. And then it circulates within all the stuff here is a subarachnoid space, okay, just below the arachnoid mater, and gets reabsorbed at these villi. Okay, so produced in the choroid plexuses, gets reabsorbed in the villi into the dural sinuses. And you should be able to know the basic flow of cerebral spinal fluid and its function. Seriously? Asshole. Was it the numbers?
Yeah, yes. You're adding each individual number.
Oh my God. What a pain in the ass. This is the danger of borrowing slides, is that you go, oh, this is great. It shows exactly what I want to show. I modified a little bit. Well, I did check for animations. Thank you. All right, this is the next slide, right?
Yes.
Okay, Jesus. All right, so what we're going to talk about today is the brain, and we're going to just do a brief overview of the different structures and kind of what they do. And so it's divided into these 4 parts, okay, where the cerebrum would be this area, diencephalon would be here, brainstem here, and cerebellum. All right, as far as cerebral structure goes, this is your cerebrum. This is your cerebrum on drugs. Back in the 80s, they had this campaign, anti-drugs, where you guys seen this? The egg, where they say this is your brain, and it's an egg. And then they break it, and it's in a frying pan, and go, this is your brain on drugs. And then, any questions? Like, did they? Still? Cool.
What about the drug spiders?
Do what?
The drug spiders.
I don't know about those.
You'll see soon.
Okay. So, these are our cerebral hemispheres, right? We have 2, left side and the right side. Right? So this is what we're talking about, the cerebral hemispheres, specifically what I'm holding, what you see, okay? And these cerebral hemispheres are divided into lobes, and the lobes are named according to the regions of the skull that they lie underneath. So the frontal lobe would be underneath the frontal bone, for example, okay? You can't really see, I mean, you can see it, but I can't tear it apart. Between the cerebral hemispheres, we have something called the corpus callosum. Okay, and that's this area right here, which is a bridge of axons that connects the left hemisphere to the right hemisphere. And that corpus callosum is a very important structure that allows the left side of our brain to communicate with the right side of our brain. If we don't have that, there's some weird things that go on, and we'll talk about that as we go through the rest of this lecture. Okay. Brains are bumpy, right? All the bumps are called gyri. The grooves between the bumps are called sulci. And there are names for every gyrus and every sulcus. You don't have to know those. These are the details that get left out, okay? Why is it bumpy? Why is it so convoluted?
More space, surface area.
More surface area. So the more surface area you have, the bumpier brain, the theory is, the more intelligent you are. However, is that always true? No, because if you're never taught how to make those connections or do anything with them, they're kind of useless, right? But there is somebody in the cohort 24 who always says that she has smooth brain. And smooth brain, without bumps, less intelligent. And people have been born with smooth brain. They don't die, they don't live very long, okay? Not able to form synaptic connections, not able to make those connections within the brain that people without smooth brains are. That condition is called lissencephaly. All right? So, bumps and grooves, know what they are. Different lobes, frontal, parietal, occipital, temporal. You also have a 5th lobe called the insular lobe, and that's beneath the frontal lobe and the temporal lobe, and any information about that is within your anatomy lecture, so I'm not going to go into any details about that. All right, now I find it fascinating that your cerebral cortex specifically, even though we call all this the cortex, what's at the center of it? No? What's just the cortex itself? What's at the center of it? How do you get information to the cortex? Axons. So what's most of the cortex made of? Axons going up to a very thin layer of gray matter. And that very thin layer of gray matter is the cerebral cortex. Okay, the rest of it is white matter, axons going up, making those connections from lower regions of the brain. Okay? That's how you feel, right? Slam brain. All right. Now, even though the lobes allow us to get a rough map of what things are, we divide it more specifically into smaller functions to make things make more sense. Now, I said all of the grooves in sulci, all the gyri and sulci have names. I'm only going to hold you responsible for one, and I'm going to argue that that's the most important one, and that is the central sulcus. Right? And why is that the most important? Because one, it divides the frontal lobe from the parietal lobe, and it's the division between our motor cortex and our somatosensory cortex. And by somatosensory, we mean all of our general senses, feeling stuff. Okay? From our skin, from our organs, things like that. Okay, so central sulcus, motor cortex is anterior, sensory cortex is posterior. And we'll get into more details about those as we go on. Other areas, this area right here is called the prefrontal cortex. In other words, before the frontal cortex. Okay, so if our frontal cortex is associated with motor, this is where all the planning of motor activities occurs. Okay, including planning how we're going to use words, right? And that would be Broca's area. Behind that, we have the parietal lobe, visual processing in the occipital lobe, which you guys told me. Between those, we have spatial coordinates of body and surroundings. So what this area allows us to do is take visual information and general sense information and auditory information from the temporal lobe and put it together to give us a picture of what's going on in the world around us. Okay? One of those things is language comprehension. So these are two areas you need to know, Broca's area and Wernicke's area. Broca's area, notice that's associated with motor, so that's our ability to speak words or find the right words to say, and they come out. Whereas Wernicke's area is language comprehension. In other words, our ability to understand written, spoken language and convey that into meaning in our brains. All right, yes?
For Broca's area, is it more so the plain English words, or is it what sends it into like actual like motor functions coming out of like out of the mouth? The motor functions.
Both. So here in Wernicke's area, that's our ability to understand information, process it, and we have connections to Broca's area that allow us to use that information to speech. To produce speech. So mostly motor, mostly coming out, but it does have those association areas as well. All right, prefrontal cortex. So this area right here that I have circled. Here are the functions. Think of it as our thinking area. I don't know if you guys have ever done this. I'm weird. You guys know that already though. If I'm sitting there and I'm reading something, and like I'm just not really paying attention, you guys can all relate, right? This is how we read textbooks. And then I'm like, oh crap, I need to pay attention. I can literally feel a shift in my frontal lobe. Like it's a weird phenomenon. Like it's something I can actually detect that's going on. And so the prefrontal area is our ability to pay attention, to think about things, to organize thoughts and perform some sort of action, whether that action be motor or whether that action be putting into memory stores. Okay, so I'm not going to read this for you, but prefrontal area, thinking and association. One thing, correlate widely divergent information. I need to be to class by 8 o'clock in the morning. To do that, I need to make sure I get up, take a shower, get dressed, get my keys, get in my car, put on the podcast that tells me the news, drive here, turn on the anatomy table, leave the door open, all that stuff. Widely divergent information. Prefrontal cortex allows us to put all that together for a goal. My goal is to get here for you guys. Okay? All right. Now, with the cerebral cortex, 95% of the human population, that means across the world, is left hemisphere dominant. Now, this is not a left brain versus right brain person. That is complete bullshit and has been proven to be wrong over and over and over again. There is no left brain and right brain person. But she can be left hemisphere dominant and right hemisphere dominant. So only 5% of the world's population, right hemisphere dominant. Our left hemisphere is where language-related activities occur. So Broca's area and Wernicke's area are far more developed in the left hemisphere than they are in the right hemisphere. Why? Because the left hemisphere is language dominant. Okay. Left hemisphere. I have a video. I actually opened the last anatomy module today just before class. There's a video in there where this woman who had a stroke, she wrote a book that documented her stroke as she was having it. She's a little crazy, but she talks about left brain versus right brain. Okay, left brain, analytical, thinks in lists, allows us to plan, organize. Right brain, a little more esoteric, like artistic, music, sensory feelings, allows us to experience the world. Here's how we operate in the world. Here's how we think about the world. Okay? So very different sides. However, we use both, right? What allows us to communicate between those two? The corpus callosum, okay? The question I usually get is, are lefties right brain dominant? And the answer is no. There's no evidence to support that either. All right. I'm hot. We're on a different time scale, aren't we?
Yes.
2:30 is an hour.
Yeah.
All right. Let's first talk about the motor areas of the cortex. You guys ever heard of that motor homunculus, where there's like a map of the body on the brain? Okay, it's really weird. It's creepy looking, right? But what that shows you is that different parts of the motor cortex are associated with different areas of our body. So in between the cortex, like if we look down on the inside right here at the motor cortex, that's associated with our lower limb, okay, and our pelvic region. As we go around to the outside, hips, legs, feet, trunks, arms, neck, lips, mouth, things like that, okay? So that's how it's organized, that's how it's mapped. Just in front of that, before the prefrontal cortex, we have these supplemental motor areas that allow us to use the information to do specific skills, or rotate our head, or do contralateral eye movements, okay? So these supplemental motor areas take the information from here and allow you to do very specific things. So that's where the actual learning occurs. When we're babies, we have all of this right here. We can control those. It's here where we start developing the useful control of those areas. Okay, our brain is arranged in layers. Okay, so if we take that really thin cerebral cortex and put it under a microscope, we can see different layers within there. The spinal motor neurons within this motor cortex are located in layer 5. So they're deeper within the cerebral cortex. Okay, they're made up of these things called giant pyramidal cells, which is why they're called the pyramidal tract. Excuse me. And you need to excite about 50 to 100 of them in order to get any useful muscle movement. Okay, so it takes a large amount of input to be able to move a muscle. And that's coming from different areas of our brain, the association areas, to accomplish that. All right. Another name for that pyramidal tract is the cortical spinal tract, and we'll talk a little bit more about that as we go on. Those cortical spinal fibers, they will always synapse with interneurons. And what do interneurons allow us to do? First of all, what are interneurons by definition? What does interneuron mean? Between neurons. So they are smaller neurons, usually located within gray matter, that allow longer axons to communicate from one area to another. Okay, so interneurons allow us to modulate control. Okay, those motor signals coming from that 5th layer will go down a direct pathway called the cortical spinal tract. And the reason why I have, does anybody know what this is right here? Right, pons, medulla oblongata. All right, this, do what? This would be the anterior side, so from this side. Right, these are the pyramidal tracts right here. All right, what the direct pathway, pyramidal tracts, cortical spinal allow us to do are discrete, detailed movements. So my ability to go up here and pick up this brain and twirl it around in my fingers requires detailed, discrete movements, right? And it's not working right. We also have an indirect pathway that goes through the basal ganglia and or the cerebellum and or the brainstem to allow us to modulate activities as well. What we're going to be talking about is the direct pathway. All right, we're going to leave the indirect pathway alone. So here's what the direct pathway looks like. Okay, you have, let's say, what would this area control, roughly?
The leg, the hip, or the trunk.
Maybe legs, hip, trunk. All right. And more down here?
Face, tongue, or larynx.
Okay.
I just flipped up the picture.
Okay. It's a good picture. Like, it's creepy, but it's good. It works. Good. So all that motor information goes down. Here's all your white matter. So when I talked about this thin layer of gray matter, that's all this pink stuff right here, but this less pink stuff, white stuff, that's all your axons. So it's coming down as well as going up through the brain. It gets to the brain stem, and then once it gets to the pyramids of the medulla oblongata, it does something interesting. It crosses. All right? That crossing over after the pyramids of the medulla oblongata is called decussation. Okay? That's an important word to know because it will appear in your exam. And I don't want people coming up to me and going, what does decussation mean? We never covered it. Crossing over. Okay? We decuss. All right. So What that means, if we have motor information coming from our left hemisphere and it crosses over and goes down our spinal cord on the right side, what does that mean for movement? Right. Left hemisphere controls right movement. Right hemisphere controls left movement. All right. Those fibers running down from the cortex through the medulla and the spinal cord, those are called motor neurons. All right? And we have two types of motor neurons. One is upper motor neurons, right? And this is going to be important when you get to your clin med, because they talk about upper motor neuron diseases and lower motor neuron diseases. So you should know what they are. All right, upper motor neurons, they start here in the cortex, they go down, and they're contained entirely within the central nervous system. So they're either in the brain or completely in the spinal cord. Okay? They don't leave the spinal cord. They're in the spinal cord. Where are they? In the spinal cord. Okay. Do they leave the spinal cord?
No.
No. However, notice that within the gray matter of the spinal cord, what do? What's happening right here? A synapse. All right, there's synapsing within the gray matter of the spinal cord, and then we have another motor neuron leaving. Is that motor neuron that's leaving an upper motor neuron? No. What would we call that? A lower motor neuron. Good. Okay. So upper motor neurons completely within the spinal cord. Lower motor neurons are the ones that are coming out. And if you have damage to an upper motor neuron versus a lower motor neuron, you're going to have different manifestations. All right, that's not a discussion for now. That's a clin med discussion. Okay, but you should know where each of them are. All right, and most of our upper motor neurons, since we're on this slide, are going down the lateral tract, lateral corticospinal tract. I don't have lower motor neurons? Huh. Good thing I explained it, right? All right, here's our motor homunculus, all right, which Jesse already had pulled up on his computer and is answering questions. Good? That's good. That's good. I'm glad you're doing that. What we've talked about so far is the motor cortex. We're in the middle, feet and toes, and then we go progressively up our body as we go out. Our sensory cortex, where is that located in relation to the motor cortex? Posterior. It's posterior to the motor cortex. Good. Follows the same general map. Okay? The difference is it starts with the genitals because we have sensory feelings in our genitals, but we have no motor control in our genitals, right? That would be really weird. Okay? But then it follows the same map up here. Okay, all the way across. What would happen if we had the sensory cortex for our feet located between our hemispheres, but our motor, did I say sensory?
Yes. Yeah.
Okay, in between, but our motor cortex for our feet located way out here on the sides? Would it be very efficient for them to communicate? No. So the reason why they're located right next to each other is because whatever you're feeling here will translate into a motor output there. Okay. Corpus callosum. Is that within the cerebral cortex? Here's its location. No, it's below the cerebral cortex. So that means all of the axons coming from the cerebral cortex have to go down towards the diencephalon in order to cross over. Okay? So, but it's important because it connects those two hemispheres. If we cut that corpus callosum, what do you think will happen? Will the person die? No. There's a video out on Canvas for you guys to watch that shows this guy who had seizures, had his corpus callosum cut, and then he did all kinds of experiments on him. He was a willing participant. But you can see some of the manifestations of that if you happen to cut it. Now, important, dominant hemisphere understands spoken word, right? And that's where we speak spoken word. Non-dominant hemisphere understands written word. So what would happen if we have the corpus callosum cut, we write something with our left hand, but we had no idea why we did that? That's what would happen with the corpus callosum cut. Or you saw something and you couldn't express it in words. Okay?
Like the word salad, right?
Do what?
Like the word salad?
No.
No.
Mm-mm. So if you want to see the manifestations, watch that video. It's older. It's like, it's hard for me to tell, 80s, 90s, but it's still a good video that shows you the importance of the corpus callosum. They literally cut it right down the center. All right. So that's all the cerebral cortex. All right. Now we move on to this middle region that's between the brainstem and the cortex called the diencephalon. All right, what does di mean? Two. Encephalon, brain. Two brain. Because there's two parts to it for each hemisphere. All right? Mostly composed of gray matter. What does that mean? Cell bodies. So what's happening there? Connections. Right? That's where connections are being made. All right, here are the 4 parts, and we'll go over those as we go along. Thalamus. All right, the thalamus is the big round part. This doesn't do it justice, but it's this part right here, right in the center. The most superior part, think of that as a central relay station for incoming sensory impulses. So all the impulses coming, all the axons coming from sensory neurons in your body, so all those upper motor neurons, no, they wouldn't be motor neurons, would they? Be sensory. So all those sensory neurons coming from your body travel up and they go to the hypothalamus first. So they make a bunch of connections in the hypothalamus. And then the hypothalamus tells it where to go from there. Okay, so it's the hypothalamus that kind of serves as, you guys ever seen this before, switchboard operators? Way back when telephones were first invented, you didn't dial. You picked up the phone and you said, hey, I'd like to call Henry Joe. And they would go boop, boop, and connect you to Henry Joe. That's what the thalamus does. They did a skit on this in Saturday Night Live back in the 80s. I put that on Canvas for you guys to watch as well. By the way, speaking of Saturday Night Live, I did see your video. All right, hypothalamus. What does that mean? Below the thalamus. So if this is your thalamus, this would be your hypothalamus. And we talked a lot about the hypothalamus with endocrine. It's a big player in endocrine control. All right, but what also does is it is where most of your set points in the body are held. Your blood pressure set point, your temperature set point, your heart rate set point in the hypothalamus. So that is its function, is to regulate those specific body functions at a certain level. All right, in the back of our thalamus, so here's our thalamus, we have something called the pineal gland, or pineal gland. This is responsible for circadian rhythms, so our sleep-wake cycles. And it is directly connected to the amount of light that your eyes are receiving. So the process behind this is during the daytime, you have light hitting your retina, sending nerve signals back. One of the areas that gets those nerve signals is the pineal gland. Pineal gland is shut off by light. But when it gets dark, let's say you're camping, because we never really experience dark in, first world countries. So you're camping and it gets dark. You have less light going into your retina. That activates the pineal gland and it starts releasing a hormone called melatonin. And what does melatonin do for you? It makes you sleepy. That shit don't work for me. But it makes you sleepy. All right? And you fall asleep when it's dark. So that's why we are tied to dark, falling asleep, wake during light period. You fall asleep, sun comes up, light hits your eye. Even though you don't have your eyes open, you're still getting through this thin layer of skin, right? Okay? Tells your pineal gland, hey, there's light coming in, wake up. This is one of the reasons why it's really bad to look at your phone at night. Because it keeps this thing telling you that it's daytime. All right. Let's finish the diencephalon. No, let's finish the limbic system, and then we'll take a break. All right. The limbic system, it's a real pain in the ass to find a good picture that shows you where the limbic system is located. Put it to you this way. It's surrounding where if this is your diencephalon, it's the area immediately surrounding it. Okay? Structures that are immediately surrounding it. All right? Limbic system, they often call this your lizard brain. It's involved in emotional responses. So, the best way to put this is, I'll give you an example for myself. I used to have a dog. We got rid of the dog because we realized it wasn't fair to the dog. We were never home. But this dog, when it was a puppy, of course, made messes in the house. All right? When it makes messes in the house and you come home from a long day of work and you had school, what's your first reaction? Motherfucker! All right? That's an emotional response. Now, if you didn't have your cerebral cortex, what would happen as a result? You'd probably go beat the shit out of that dog. Okay? Because you have this intense emotional response. But because we have a cerebral cortex that can override the limbic system, that can guide us into behavior that doesn't allow us to do that. So the purpose of the limbic system, if you look at an evolutionary timeline, is to increase our chances for survival. Like, we're going to do things that we wouldn't normally think about because it's going to help us survive. Now that we live in society and we're supposed to be societally appropriate, we override those systems. Okay? Also, this is involved in the sense of smell of the limbic system. There's a little area right at the front called the amygdala that's associated with the sense of smell. And we'll talk about that when we get to the sensory system. All right, let's take a break there, and we'll start again. All right, limbic system. We did that, right? Hippocampus. Do you guys recognize this? Where'd this come from? Anatomage. Actually, this actually.
That's creepy. That's awesome.
It is.
The eyes. They're creepy.
Okay, have you seen Attack from Mars? Is that the one?
Yes.
Okay. It's really old, but if you
Yes.
know,
Yep.
it came out a year ago, but it's like
I would recommend, if you guys are really interested, do it only if you have time, and find out where the limbic system and all these subcortical structures are. The anatomage is gives you a really good 3D way to look at these things. And so this is what I did. This actually comes from a Clin Med lecture that I gave on neuro, and I decided to put it in here. What we want to focus on for this one is what's in yellow. So this yellow part right here is called the hippocampus. All right, and the hippocampus is important for memory. Okay? All kinds of memory. All right? But we're going to talk about the specific function that it does is what's called consolidation of memory. And by consolidation, we mean converting short-term memories into long-term memories. Because if we don't have the hippocampus, if we have damage to it, we can't convert short-term to long-term memories anymore. And what do we call that? Pardon? Who said it? I thought I heard somebody say it. Dory. Anterograde amnesia. We can't form new memories. So everything we have is all in the past. Movies that address this, 50 First Dates, Memento. I don't remember 50 First Dates, so I couldn't tell you. But I know it addresses it. Memento, has anybody seen that movie? That's a really good one. If you're a fan of, what is his name? Christopher Nolan. If you're a fan of Christopher Nolan, this is a really good movie. And Finding Nemo. Although if you really, really know about the hippocampus, I just wanted you guys to get freaked out by the picture again. Trauma. If you really pay attention, it's not true. Anterograde amnesia, if you watch these movies and pay attention to what they're saying and what they're talking about. I mean, it's artistic license, right? All right. So hippocampus, we know it's important for converting short-term to long-term. If we don't have hippocampus, anterograde amnesia, can't form new memories. I think I might have a video out on Canvas as well that shows a case of anterograde amnesia. If I don't, I will put it out there. So in doing so, the hippocampus determines what sensory experiences are important and which don't require attention. So, you guys ever done those things, these videos, where they say, pay attention to this, like something's gonna happen, and you're like, and everything around you, like, you're not paying attention to it anymore. Then all of a sudden, like a scary face pops up and scares the shit out of you because you're paying so attention. So, it's, do what?
They see the gorilla was there the whole time.
Or that video. You guys ever seen that video? Okay, where they're bouncing basketballs around and you're paying attention to which person the basketballs are going between. But in the middle of this gorilla comes in the middle, dances around, and then leaves. And you never pay attention to it. So it's the hippocampus that's responsible for you selectively divvying up your attention. All right? Most wives would agree that men have a defunct in their hippocampus.
This might be
Defect.
too deep, but when you have like trauma and you like forget, like and your brain's like protecting yourself, is that what the hippocampus is doing, or is that okay?
It's definitely involved, yeah. Or you selectively, like God, I'm not thinking of the word, where you go someplace else in your brain and you, hey, what? Dissociate, yes. You dissociate.
We're all familiar with that. All day. Yeah.
All day. Okay, now the thalamus, remind me again what the thalamus does. Switchboard, relay station, right? So it turns out that the thalamus is also important for recalling memories because it takes sensory information, plugs it into different areas of the cortex, and brings it back to say, oh, that's what that was associated with. Okay? If we have damage to the thalamus, because long-term memories are stored in our cortex, not in our hippocampus, then what we have is retrograde amnesia. We are unable to recall distant memories. So we can form new memories, but we can't recall distant memories. Okay? So We'll just leave it there. So what is memory? The answer is we really don't know. We still don't know for sure. But there are some things that research has shown us that's true about memory. So first we'll talk about it at the macro level, which it means when you're a provider, how can you test different types of memory? And then we'll talk about different mechanisms of how this might occur. So immediate memory. If you're with your patient, you want to test immediate memory, give them 3 words: dog, cat, ball, and say, repeat those back to me. Dog, cat, ball. Okay? That would be immediate memory. Short-term memory would be, all right, I want you to remember the 3 words, dog, cat, ball, and then I'm going to lecture about memory, and then I want you to repeat those words back to me after I lecture. Okay? If you can't repeat those words back to me, then you have a problem with short-term memory. And where's the nature of that memory held? Probably hippocampus.
So the short-term memory example you just gave, when they test for concussions, like in the SNAP 5, they do something similar like that. Is there like, are they testing for damage on like the campus, or
Yeah.
that's what?
Mm-hmm. And then long-term memory, you would ask your patient something that they can verify, like, who's your wife? Who was the president? Who were the presidents in the 1980s? Long-term memory. Okay? Or something like that, okay? So something long-term, but it has to be verifiable. You can't just ask them a random question. They could say anything, right? And you wouldn't believe them. You have to be able to verify that. So you talk to their significant other, for example, and say, hey, is this true? Or give me something that I can test them on.
So problems with all 3 of those would deal with the hippocampus. Like if you had a problem with any of those, it would be the hippocampus.
Not necessarily, because remember, long-term memory is held in the cortex, and if you have damage to the thalamus, you can't recall those memories. So that would be either cortical damage or damage to your thalamus. All right, immediate memory, mechanism for that, accumulation of calcium in the presynaptic membrane. So you fire a neuron down a pathway, you have calcium being released in here, and calcium causes the release of vesicles, right? So that increase in calcium, release of vesicles, you're continuously stimulating that pathway for a short period of time. Okay, you guys ever like ask somebody for their, okay, you don't have your cell phone with you. They say, what's your number? And you're like, all right, 419, and you keep repeating it over and over and over again. This is what you're doing, is you're constantly stimulating those neurons in that pathway so you remember it. And then as soon as you put it in your phone, boop, gone, and you never remember it again. All right, short-term memory may result from temporary physical or chemical changes in pre- or postsynaptic membrane. So here's your presynaptic membrane, here's your postsynaptic membrane. If you continuously stimulate a pathway, you may put more proteins in the presynaptic membrane that will allow for vesicles to dock more efficiently, or you may put more neurotransmitter receptors in the postsynaptic membrane, which will allow you to respond to continuous stimulation much better. All right, so that's proposed mechanisms for immediate and short-term memory. All right, so that repetitive stimulation that we talked about, let's draw the neuron again. We continuously stimulate. We have a progressive decline in sensitivity. We call that habituation. A good example of that is I put my watch on in the morning. By the end of the day, I don't even feel it anymore, right? I don't realize that I'm wearing it. Okay? The reason for that is the amount of calcium channels here that are open to allow vesicles to be released declines. All right, so if you don't have as much calcium going in, no matter how many times you stimulate, you're not going to have as many vesicles released. So we're not going to feel it, for example. All right, now if we have a neuron within this pathway called a facilitator that will activate this neuron as well, increase its responsiveness, then that can prevent habituation. So an example of that would be, I have my watch on, I forget about it by the end of the day, but let's say I set it to vibrate every second. Am I going to forget that watch is there? No, because I'm constantly stimulating it and I'm preventing habituation because I'm activating those facilitator neurons. So that's all that this is showing you. This thing right here is what I just drew on the previous slide. Okay, that takes place through internal cell signaling, right? Which I'm not going to ask you about G proteins or phosphorylation. Internal cell signaling is what allows this facilitator neuron to keep this active and send signals, all right? So the point is, if I have that noxious stimulus telling me my watch is there every second, I'm going to remember my watch is there. Okay, so when I do one of these maneuvers, I don't hit something and forget it's there and break my watch. Which, if you look at my watch, it's got all kinds of scratches and dents all over it. All right, long-term memory, what they think, and one of my postdocs was doing this in a laboratory, looking for synaptic changes in long-term memory. What they think happens is that you have long-term structural changes in the synapse, which strengthens it. So if you continuously stimulate a pathway, you're going to induce changes between the presynaptic and postsynaptic membrane that are permanent. And that's what allows you to form that long-term memory. Okay? If you don't use that pathway, you start to lose that tight connection. So very much a use it or lose it type thing. For example, let's say you learn how to ride a bike as a kid, right? You don't forget that. And then for some reason, you never ride a bike again until you're like 50 years old. When you first get on that bike, are you going to have some problems? Yes. That's because some of that synapse got weakened. But as you pick it up and facilitator neurons stimulated, it will allow you to reform that connection. Okay? So permanent changes in the synapse are what we think happens with long-term memory. Okay? Now, in order to convert short-term to long-term memory, we have to have repetitive stimulation of the same pathways, which is why it's important to not just come to lecture, but to repeat that information in your studying. Okay? Because the more that you repeat that information in your head, the more likely you are to make strong synaptic connections and increase your conversion from the hippocampus to long-term memory centers in the cortex. And of course, that requires time. We don't all remember the first time, right? Some things we do. Most we don't. All right, basal nuclei. Those are also subcortical structures. Within our white matter, there are islands of gray matter that form our basal nuclei, and these are the three structures that are associated with it. These structures produce dopamine. And remember back a few slides when we talked about motor pathways, we talked about the indirect pathway that goes through the basal ganglia. This would be part of the indirect pathway. what these areas do is their neurotransmitter is dopamine. Dopamine is an inhibitory neurotransmitter. So, let's say this is our synapse, okay, and this is releasing dopamine, and it binds to receptors. What kind of channels are you probably going to open if you're inhibiting the next neuron? Potassium, right? So you're likely opening potassium channels. Potassium leaks out, and what happens to the potential of that next membrane? You get more negative, hyperpolarized, okay? Because you've opened up those potassium channels and allowed potassium to go out, right? What that allows you to do is it inhibits excessive movements, okay? People with Parkinson's disease have problems with their basal nuclei, right? And what are the manifestations of Parkinson's? Right? That's excessive movement because of damage here. If we give them dopamine, for some patients, not all patients, that will lessen their tremors, okay, and restore some of that function. All right, now we're in the home stretch. The brainstem, all right, the lowest part, right here, that allows us to communicate from the higher brain centers to the spinal cord. All right, brainstem has sensory and motor, as the spinal cord does, and the brainstem is also where most of our cranial nerves come from. All right, so once we get to the cranial nerves, they'll come out of specific brain centers to perform specific functions. What's the one cranial nerve we've learned a lot about already? Vagus. Sweet. Which is cranial nerve what? 10. Awesome. All right, 3 parts. Midbrain, pons, and medulla oblongata. All right, in order from superior to inferior. So we'll talk about them in that order as well. The midbrain, we look at the anterior part of the spinal cord, it has this area called the corpora quadrigemina, or 4 bodies. Okay? That's divided into our superior colliculi, left and right, and our inferior colliculi. Superior colliculi are involved in visual pathways and visual reflexes. So I see something out of the corner of my eye, and I duck. Okay? That's a visual reflex. Or, you ever played those games, or I used to have a running watch, and we used to play this game where like you press start and stop really quick to see how quickly you do it. Okay, that's a visual reflex. Auditory then would be I hear something, I turn my head. Okay, that would be the inferior colliculi. So superior is visual reflexes, inferior is auditory reflexes. All right, these are connected with the reticular formation. We'll talk about that at the end and discuss the importance of it. The next area is the pons. We covered that when we covered the respiratory system because it has the pneumotaxis area, which is responsible for our breathing or helps us with our breathing. And then finally, at the bottom, we have our medulla oblongata, which is, as you know, regulates a lot of our autonomic functions. Okay, so that's where our cardiac control center is, which sends sympathetic impulses to the heart to control rate and strength of contraction. Vasomotor center, which, of course, we know vasoconstricts. Respiratory centers, and also some non-body reflexes, some of which we still don't know the function of, like hiccups, sneezing, things like that. All right, the medulla also is the location of our pyramidal tracts. And what are pyramidal tracts associated with? Corticospinal, coming from where? What part? Which cortex? Motor cortex, good. Coming from the motor cortex. And just below that, we have an area where they decussate, which means what? They cross over. So another important function of the medulla oblongata is that it has our corticospinal pathways that decussate. All right, now the reticular formation is kind of complex and not 100% well understood, but this is where you would find the reticular formation, and notice that it's associated with the midbrain, the pons, and the medulla. All right, so it interacts with all three of those areas. And one of the things that we think that this does is it tells our cortex to wake up. Okay, and that's based on incoming sensory input. So, what helps us get to sleep usually? A nice, quiet room. Or if you're my wife, she has to have every fucking fan in the house on, making noise. But that is habituation, because you're used to that noise, so it doesn't become sensory input anymore. But, so you're sleeping, nice quiet room, or you're habituated to the fan, and then all of a sudden, you hear your kid screaming. That's sensory input. Okay, that's going to go through the reticular formation, and reticular formation is going to say, hey, we've got something going on. You might want to wake up for this. Okay, and what is the purpose of that? Survival. Good. Survival. I wish. So I track my sleep every night on my watch because I have had problems sleeping since I was a little kid. And I looked last night and I got 4.5 hours of sleep. And I know some of you can relate because some of you told me I only got 2 hours of sleep last night. I'm with you. I feel you. Right on. All right. The sleep deprived. The zombies. Speaking of zombies, did anybody get to watch the fireworks at all? Any fireworks? Okay. There's a movie. It's one of the Dead movies. And if you've seen this, you know what I'm talking about. One of the ways they distract the zombies is fireworks. And they're like,
It's like
okay. So, one of your IOs talks about different states of consciousness, which is why we're talking about sleep. If we have a state of unconsciousness and we can be aroused from it, that's sleep. If we can't be aroused from it, that would be a coma. Okay? So, I want to mention sleep. I'm not going to go into a whole lot of detail about it, though. 2 types of sleep, slow wave and REM. Okay? Again, very brief overview. So if you're like, that's not how sleep works, surface. Okay? It is important. So slow wave sleep is that deep sleep. My watch tells me in the morning how much deep sleep I've had. And when I looked at it at first, I'm like, it said that it was good, and I only got like 2 hours of deep sleep. I'm like, what the hell? Like, that's all I need? But if you look at the graphs of this, that's about all you need for a good night's sleep. Is that would be quality sleep, not amount of sleep. Okay? Slow wave sleep is that restful sleep, that deep sleep, not associated with dreaming, associated with decrease in breathing, respirations, and heart rate, and not associated with dreaming. But dreaming can happen in that state, but usually we forget about those. Rapid eye movement sleep is a different sleep. That's when we are actively dreaming. Those are those dreams that we remember. Usually peripheral muscle tone is inhibited, so your brain is really active. Your eyes are darting back and forth, but your muscles are kind of limp, so you're not really going anywhere. Brain waves during REM sleep are similar to wakefulness, all right? And we're not 100% sure why we have REM sleep, but we do know that the brain waves are similar to wakefulness, and those are we'll get into it, beta waves. All right, and REM sleep occurs in cycles. And if REM sleep occurs in cycles, so does deep sleep. All right, so the way it works is this. We're awake. All right. Alert wakefulness, beta waves. So alert wakefulness, we see beta waves. Those are our fastest waves. We're sitting here reading a book. We are engaged in a conversation with somebody. We're watching a movie we're really engaged with. Okay? That's beta waves. That's alert wakefulness. Quiet wakefulness, which is one of the reasons why they say meditation is so good, is sitting there with your eyes closed, just being really calm. Okay? Those are alpha waves. Okay? So alpha waves kind of start occurring when you're going to sleep. And then notice that you go into deeper stages of sleep. But if these represent hours, how long is that first cycle of deep sleep? Hour and a half. It's going from here to here. I would say an hour or less. Okay? And then you go up into REM sleep, and what does that last? Maybe 30 minutes, less than that. Then you go back down, about another half hour to an hour of deep sleep. So if you add all those up, let's say each one is 45 minutes. That's an hour and a half. That's 3 hours. That's 4 hours and 25 minutes. So you don't need a whole lot of deep sleep. But the more deep sleep, but if you get like yes, you don't need a whole lot of deep sleep, okay? You only need a little bit. So when I go to my watch and I say, oh, I've only had 2.5 hours, and it says that's good, now I'm like, oh, okay, that makes sense. Because if you look at the cycles, they will corroborate that. All right, why do we sleep? I'll just give you the broad answer. We don't fucking know. Okay, we really don't know. The mechanism, how we go to sleep, how we stay asleep, and how we go between areas of sleep is not really known. I'm not going to test you on the details of sleep. Okay, that picture before, REM sleep, deep sleep, the cycles, the basics of that, okay, but these slides, not really so much. Okay. So let's just, I'm not saying cross them out, read for your own pleasure, but I'm not going to test you on this stuff. Okay. Again, REM sleep, function unknown. But what kind of waves are produced in REM sleep? Beta. And that's similar to what? Wakefulness. So that level of understanding. All right. We also don't have explanations for sleep-wake cycles, except for that, they're related to hormones and light. But there are theories. I'm not going to test you on those theories. And what about what sleep does for us? Well, for the body, it doesn't do a whole lot. Like, rest is important, not doing anything, but sleep itself, not really important for our body. But it is profoundly important for our brain, all right? If we don't have sleep, you guys ever seen this? It's a YouTube video, and supposedly they did this study in Russia where they deprived these guys of sleep, and it's if you actually go and look at the details of that, it's not real. That study was not real. But what they do know is that if you do deprive people of sleep, it does cause psychosis. Not to what the level that they describe in that video, but it can cause psychosis and altered mental states. My wife tells me she knows I haven't been getting good night's sleep because I'm irritated all the time. Okay? But what we think happens is it basically resets our brain and it allows us a fresh slate to deal with the next day. All right, brain waves. If we record brave waves, if we record brave waves, what do we call that? We hook electrodes up and we record them using a what? EEG, which means what? Good, electroencephalogram. Okay. So what brain waves are, what we're recording, are firings of groups of neurons in a coordinated manner. Okay, and that's important. What produces these waves are groups of neurons firing in a coordinated manner. And we've recognized certain patterns of waves during different activities, if you will. Okay? So you should be aware of these waves, where they come from, and what they are related to. So I'm going to start one with beta waves. Why? Because beta waves theoretically are what you should be in right now, because you are awake, completely engaged in this lecture and thinking about how great the brain is and worrying about your exams. Right? Okay, those are beta waves. Intense mental activity or stress. 2, alpha waves. So those are the fastest. Alpha waves are the next fastest. All right? Remember, alpha waves slower. You're awake, but you're resting. Okay? Usually your eyes are closed because as soon as your eyes open, what happens? You're paying attention to what's around you and you're in a wakeful state. Okay? Alpha waves, mostly in frontal and parietal regions. Okay? Mostly in the frontal parietal regions is where they come from. They've done experiments where if you're sitting there with your eyes closed and you're producing alpha waves, and then you open your eyes, immediately they switch to beta waves. Then you close your eyes again and they switch to alpha waves. So it can be that quick, okay? And doing that can be recorded in electroactivity. If you're ever interested in the research behind that, it's fairly interesting. Next, theta waves. So that would be third fastest, right? If you remember from physics, because I know you all love physics, hertz means how many per second. So the higher the number, the faster it's going. The lower number, the slower it's going. Theta waves, they originate from parietal and temporal lobes, particularly in children. In adults, they come from more widespread brain regions. Usually theta waves, emotional stress in adults, and response to disappointment and frustration. So theta waves. Now, I'm going to argue with this because one of the theories about theta waves is the more theta waves you produce, the more you're able to form memories. So they think that theta wave stimulation is one of the triggers for memory formation, which is probably why you remember all the negative shit that happens in your life, but not the positive stuff, right? Like course evaluations. Like, oh my God, I got 5s across everything, but that one student out of 26 said, I don't think you should be let's even go to this. I didn't like that one slide in the brain lecture that talked about brain waves. And then I'm like, oh my God, they hated me. Right? This, because we're human, this is what we do. The slowest are, just spit. Delta waves. All right, delta waves, that's our deep sleep. And if you look at it, these are the slowest waves. They occur at undulations. So those are happening in the cortex. Relaxation. Deep sleep. And there's a summary slide. And I think this is it, right? We're going to get done early. I know, but we're at like 3:16. You guys get a break. Fuck yeah. All right. Last section, cerebellum. What does cerebellum mean? Little brain. So it's that little brain sitting just below our occipital lobe. All right. Its job is mostly involved in motor functions. All right. So if you remember back to when we talked about the cortical spinal tract, we talked about that being a direct pathway and the cerebellum being involved in indirect pathways. All right. Specifically, these are general functions. Here's the anatomy of it. Like, I'm not going to go into the details of that. But you have 3 parts, right? And these are called peduncles, which means little feet. Because if you look at them, and you can't really see them on this, but wow. Maybe 3 slides will take an hour. Little feet of the cerebellum that are associated with different parts of our brainstem. Okay? What happens is, with the inferior peduncles, I'm going to go back and forth between slides. Inferior peduncles, where are we? Right here. Okay? It's receiving sensory input coming up the spinal cord. Oops. Right? Sensory input telling you where you are in space. So it will receive input like, I'm right here. You ever seen those video games where you're like creating a character and you're spinning around and they're like, where you are in space. All right. Middle peduncles. All right, that connects with the medulla. Middle peduncles, right here, connected to the pons. That's associated with connecting to the cerebral cortex about where you want to be. So I'm right here, inferior, where I want to be, middle, coming from cortex, middle, right? So here's where I am. Where I want to be is over there. Or where I want to be is not falling down. And then the superior peduncles take correcting info to the midbrain and allow you to be where you want to be. Does that make sense? So where you are, where your desired position is, and corrective actions to get to that desired position. Most of this is involved in balance. Okay? So if my inferior peduncles are telling me I'm right here, my middle peduncles are saying don't fall down, and my superior peduncles are looking at my environment and allowing me to maintain balance, that's the way it works. Okay? Because what in my midbrain is associated with my ability to keep balance? My balance. Not necessarily, not your ears. I mean, part of your ears, inner ear. Right? But also my eyes, because if I'm standing here and I close my eyes, what happens? I start losing balance. So that shows that visual input is really important for maintaining your balance, and that all that information occurs through those peduncles. All right, and the rest of the slides just showing you anatomy of it. So that got covered in the anatomy lectures. And we're done. Any questions? Good. I didn't want to answer them anyway. That's convenient.
If you have dementia, aren't you having...
All right, so I said I was going to let this run.